The second wave is upon us. Ontario and Quebec go back to lockdowns, which will cripple their economies. The need for rapid COVID testing is crucial to flattening this curve, but when will it be available? Hello and welcome to the Unpublished Cafe. I'm Ed Hand. A number of hotspots in Ontario have Ottawa, Peel Region, and Toronto coping with more lockdowns. No more indoor dining, no more working out of the gym, no more going to the theater. Several areas of Quebec are in the same situation. The federal government's announced the purchase of almost 8 million rapid tests. It was aiming to conduct about 200,000 a day across the country, but currently we're only seeing about 70,000 tests completed. The question for many is, why is it taking so long for Health Canada to give approval to new tests? Currently, about 100 are under analysis. Coming up in the Unpublished Cafe, we'll take a look at what's available now, what's coming down the pipe, and whether Canada has the lab capacity to meet the demand. Dr. Stephen Newmaster is a genomics professor at the University of Guelph, and he joins us now. And Stephen, how would you characterize the Health Canada approval process?
Good morning. It's good to be on the show. How do I characterize it? It's a very rigorous process. It's a process where they get the machines and they must validate all the evidence to make sure that it works for us and that we're not going to have any problems. So that's a fair bit of work, and it's new. It's not something we had in place before. We were all kind of caught off guard with this pandemic everywhere, and now society has to set up the infrastructure.
But it seems that Health Canada has a bit of a different approach than the U.S., as they seem to have a bit more tests approved at this point.
That's true. The U.S., the EU, they've moved very quickly. Much bigger organizations. They have much more funding. They have more staff. It's the same process that we go through in validation, looking at specific experiments and things like specificity, sensitivity, reproducibility of the results. They do share some data across these organizations at a global scale, but the due diligence is upon the country. So Health Canada must feel comfortable that that test is going to work.
You brought up funding. They have, in the EU and the US, they have more funding. Is this basically a money issue, or is it a regulation issue?
It's both. Of course, more resources, then you can do more science, and you have more people. It definitely is resource related, but it's also related to regulatory. And keep in mind, too, we didn't have the infrastructure in place, which includes regulatory protocols and validation. How do you actually test and know that these instruments are going to work, especially point of care instruments? So lab is one scenario where we test, but moving the testing into society is something we haven't had before. And it's possible. We do it in other sectors, but this is the first time we're doing it with human pathogens.
Now, polymerase chain reaction tests are the most accurate. Antigen tests, not as much. Is it because they measure different things?
Yes, it is. So there's the antibody test, the antigen test, and then the RNA test, which is a genomic test. The antigen is the protein coding around the virus. And you're doing a test, which is a chemistry-based test, to look at that protein, identify that protein that's associated with the virus. And that can be very specific at times or non-specific. So a DNA test is a hereditary test. It's a test that we use to identify individuals. It's the gold standard forensic test we use in law to be able to identify a person at a crime scene. That's DNA, and that's a different approach because you're going in and you're actually sequencing or going for a section of the genome. And for this virus, it's RNA. So two different tests. One's a chemical and one's a true genetic test. And the true genetic test, the Pcr test, is the gold standard right now, set out by CDC.
Pool testing, how does it work, and what kind of accuracy does it have?
So pool testing is when you take a bunch of samples from the population, take a bunch of people, you pool the sample and you test and you say, is there any positive for a virus? And there's complications because there can be chemistry from all these pooled samples that could impede the reaction or cause false negatives and positives. So that's one problem. It's just a strict chemistry issue with how the test works. The second problem is, okay, you get a positive from a pool, but now you don't know who specifically. So you have to go back now and do the individuals. So you could do large crowds and break them into groups and then say group A is negative, but group B is positive. And then you have to go back to group B and sample everybody and test them. So it's really just a sampling strategy.
Now, in terms of the PCR tests and the antigen tests, why is PCR so much more accurate?
So we have these concepts of specificity and sensitivity. So when we take a virus or any organism, we sequence the whole genome. We can see large sections of the genome, and we can find differences, which we call probes or single nucleotide polymorphisms, that separate that taxa or that strain of the virus from a different one. And so the specificity of PCR is really high because we go in and we know specific areas, SNPs, that will separate out one strain from another. And that's a really important concept to think about, specificity. The other is sensitivity. You can look at 10 copies in a microliter, like very small amounts of the virus get detected. So with the antigen, it has low sensitivity. You have to have a high virus load in an individual. And you don't have high virus loads all the time. You could be contagious with low virus loads. So the PCR test will pick that up. It's very sensitive. And it's very specific.
Okay. Now, from your perspective, we talked at the beginning about the approval process. I wondered, did the government stumble when it came to rapid testing or the availability of it? Or, as you had mentioned off the top, that, well, we were all kind of caught off guard by this pandemic in the first place?
No, the government had to, first of all, make sure that our clinical labs had the appropriate infrastructure and tests in place. And that was the first step in setting up a testing system for Canada. And I know there's a lot of complaints about people going into the lineup and finding clinical labs to get tested, but we've only had a few months as scientists in Canada to be able to set this up. The next stage is, okay, we have point of care, small devices like the high-risk B cube that you can hold in your hand, or the Abbott machine, which is a different test, it's the antigen test. And bringing those into society, but validating to make sure they work, and then saying, how would you use these? Where can you use them? Could you use them in airports or schools? Could you use them at mining sites? Could you use them in remote communities in the northern part of Canada, which we have? And that was the next question. And those are big, detailed questions to go through, and they've only had months to do it. So it's a huge challenge. And I know a lot of those scientists who are working, and they're working long hours, many of them 6-7 days a week.
Now, you brought up the high-risk B cube, and I know you are doing some advising for the company behind it. Can you tell us about it? It's what the high-risk B cube rapid molecular testing device.
Yeah, so first thing I want to make sure we acknowledge, like, I'm agnostic to, I have nothing to do with high-risk B cube. So we looked at a bunch of different systems in my lab, 25 different systems, and compared them all to one another. And the high-risk B cube really stood out for a whole pile of reasons. So first of all, it's very small. It's a small silver box, kind of like a 10-centimeter cube. And it runs a polymerase chain reaction, the exact same test that we set up with CDC that you run in clinical labs when you go into the hospital. And it can do it in 90 minutes, and it can run through 6 patients at a time in 90 minutes. So although we hear Abbott's really fast, and it's an antigen test, so it's not as sensitive, it's not as specific, it can only run one sample in 15 minutes, which is actually closer to 20 minutes. So they have the same capacity because 6 in 90 and 1 in 15 is really the same. So they have the same capacity machines. One's more sensitive, one's more specific. That's the high-risk B cube. The high-risk B cube can also do what's called a LAMP test, or isothermal amplification. A LAMP test is a quick test. So it's a test that runs in 26 minutes. And so you put a sample in, and it's running, so it's keeping track of all these samples. You're putting 6 in, and it's actually moving up to 12 samples, or 16 samples. So you run these samples, and in 26 minutes, you get a positive, then it's a quick test, and you want to reassure you just leave the sample in there and it runs to the 90 minutes and you get the gold standard CDC test. And there's no other machine on the market, and that's why I recommended this machine to this particular company and also to Canada to say this will be a screening test that gives you a backup without having to send it out to a lab. Just does it right there on the spot. So that's a fantastic innovation, and it's used in the food industry and has for almost a decade for food fraud to identify authenticity of ingredients and also foodborne pathogens. So it has a huge track record globally.
And is it available on the market right now?
Yeah, it is. Yeah,
Oh, wow.
Songbird has stock and are talking to companies. It's available. It's approved. It's approved for point of care, which we don't have another point of care. We have a Sophia machine, but it's a bigger machine. It's very expensive.
Do you have to be
it's
a healthcare
great machine though? Sophia's wonderful.
It sounds like it, but do you have to be a healthcare professional to do the test, or can it be done by anybody?
You need training to do
Right.
it, but I work with high school classes to test food ingredients on that machine, and also as a prof at the university, first year students, and I can train them in half an hour to use it from zero knowledge. So it's pretty easy to use. Sample prep takes 3 to 5 minutes once you have some training. So after a half hour training, 3 to 5 minutes, prep the sample. And sample goes in the machine runs. And then it has a data output that you just read on your computer, your iPad, your phone. It gives you those results immediately. And it analyzes it and puts it in whatever form you need. And the other cool part why I recommended this machine is that they're hooked together in swarms. So machines talk to each other and you can look at an output. So you could look at, say, First Nations across Canada decided we're going to use these for all our First Nations. And then you have clinicians that are serving them in remote hospitals. They can see all the results, all the outbreaks in geospace and time because the machine has a geoposition on it and it has a time card on it. You have all the results, which are the CDC gold standard. They're all connected already into a swarm within a cloud internet system that's under blockchain security. So this is something we don't even have in clinical labs where they're all connected together. So that's a pretty cool innovation.
Yeah, very much so. And it could certainly be handy right now in the middle of a pandemic. Stephen, I want to thank you for joining us and telling us more about it. That was terrific.
My pleasure. Thanks for educating.
Dr. Stephen Newmaster is a genomics professor at the University of Guelph, as well as an advisor to Songbird, which secured authorization from Health Canada to sell the high-risk B-Cube rapid molecular testing device. It seems we can't go a day without hearing about some new form of COVID testing. Health Canada has given the green light to a new rapid antigen test. And to find out more about it and the possible national strategy for testing, I'm pleased to be joined by David Junker, professor and the chair of biomedical engineering at McGill University. And David, why does it seem difficult to develop a rapid antigen test with the accuracy of PCR?
Hi, well, there are rapid tests now, and some of them actually meet roughly the accuracy of PCR, and some are in fact PCR-based. A big question about the accuracy of these tests lies also in sample collection and the sample processing. And so, if that's done in the field, that can be sometimes less or self-collected. That can be less accurate than when it's done administered by a health professional and then analyzed in a centralized lab. And then in terms of tests, so the sensitivity, we have to think which test we want to use exactly for what purpose. So if we want to have a test to really identify who is infected, then PCR test remains the gold standard, and some of the field tests are actually quite performant. If you want to detect maybe not just who's infected or not, the key aspect maybe is more contagion and infecting of others, then it's maybe a rapid test can have big advantages because it will give you a much faster response. And also you need to have a high, or it's presumed, and all data can converge in that direction. You'll need a fairly high viral load before you can infect someone else. And so rapid tests, their sensitivity actually matches, is good enough to detect someone who has active virus and who could infect someone. And so that then combined with the speed of these tests, this would then give us an advantage in identifying and isolating the people who need to be isolated.
When you talk about viral load, are we talking about people who are infected or perhaps somebody who had, if it's a lower viral load, somebody who might have been infected or been through COVID and are just in the midst of recovering?
Yeah, so you're bringing up the right point here. So, I mean, before we're infected with the virus, we have no virus at all in our bodies. And what happens once we get infected, the virus will start to multiply and grow exponentially. And where this virus is insidious is that we will have we know from some data that and from the transmission that the viral load will get very high even before we have any symptoms. And so then it's a small number of people then we have both the capacity of having a high viral load and some airborne transmission, since this is now really, it seems that it's the most effective way of transmission. So they have a high viral load and somehow this virus gets out from them. And these are the ones who then transmit to other people. And once the symptoms arise, then that's when the viral load actually starts to go down. And there have been no documented infections of people beyond 7 days of the appearance of symptoms for mild cases. And so once the virus goes down, then we can't infect other people anymore. But you can still have PCR positive tests 15 days later. And so the PCR test basically will detect people who have very residual virus, which was maybe deep in their body, but they have no maybe even active virus anymore. But so then we're spending a lot of resources on isolating these people, tracing these people, whereas maybe they haven't really had an active transmission event, or the likelihood for them having had an active transmission event in the days preceding is quite low. And the challenges with PCR testing is because the testing is kind of sparse, so we only test rarely, is that we really have a hard time of catching the right time where the person is in this active phase. And that's where then rapid testing and antigen tests in particular could be a solution, because they could be used almost for daily testing. I mean, ideally, though, the vision would be to have a daily $1 test, and then that could help you very efficiently catch when the viral load goes up and when you become infectious, and then also identify more effectively when people are not infectious anymore.
From your view, how has Canada's testing response been to the pandemic?
So, I mean, Canada had a good initial response in terms of implementing, I think, the RT-PCR testing, and we were scrambling like all the other counties to set up this big infrastructure. And to combine with the lockdowns, this was then efficient in bringing down the virus and tracing the initial infected people. Where then Canada has a bit missed the opportunities to prepare for this second wave and really consider these rapid testing approaches. And so Health Canada was really reluctant. I mean, they were I would say outright discouraging towards rapid tests. And initially, up to about a month ago or so, or a month and a half ago, where under then the public pressure, they started to consider rapid tests, but the guidelines for rapid testing have only become very recent. And so when we speak rapid tests, maybe, and we have slightly touched it, there's really different types of rapid tests. And so there's PCR-based tests that look at nucleic acids, and all of them so far, they're instrumentation-based. So you need an instrument, and you also need actually a health professional to administer you the test, so with a nasal swab. And then there's another class of rapid tests that are instrumentation-free. And so we're thinking more like a pregnancy-type test. A pregnancy test, you can go at the pharmacy, and then you can basically apply urine to it, and in 15 minutes you have a result, and there's no instrumentation needed. And so these are what's called antigen tests, or the format they're targeting the antigen, so they're targeting the proteins that make the viral envelope. And so these tests, they can then run, they can be scaled up very quickly because you don't need an instrument. And also with the possibility now that's arising of doing them using saliva, you could really also have the possibility of self-testing or self-testing at home or self-testing under surveillance. And this could then be deployed much larger scale and and again, give you the results in a much faster time of response. And so now Health Canada is in the process of approving these tests. And I think that's a very good step. I think, unfortunately, we are at the risk of being trapped in cycles of too little, too late, because now many jurisdictions and many countries start to see the potential of these tests. And of course, all countries would like to adopt these tests and deploy them, or if as they become proven or as their use becomes better established, there could be a huge demand for these tests. And since all the suppliers for these tests are located outside of Canada, we are very likely facing a supply bottleneck. And I think we're already facing supply bottlenecks basically with the current tests. And so what I think has been missing and is still missing is really a national strategy and a national mobilization towards how to tackle the testing challenge and how to ramp up the production capacity and also develop actually a Canadian basis for rapid testing.
David, that's exactly where I wanted to go next. Now, you want a national strategy on rapid testing. Why, first off, and how would it work?
So why we need? I think the reason why we need is what we're seeing now. We have a second wave. We have bottlenecks and enormous delays in terms of getting the test results. We realize that we have hotspots. We are not able to respond efficiently to these hotspots. And given that with the supply bottlenecks, I think it will require coordination and planning on where actually to deploy our resources. And so that would be just the first step in terms of with having limited resources, how can we deploy them best? Now, we also need efforts in terms to better understand. So, for example, we have still very little understanding of the viral load. I mean, we have like this theoretical understanding of the viral load increasing, but we don't have experimental data for that. So we'd also need to collect this data then to understand when the virus is starting early in people and the time course of that. And for example, for this, we need someone to actually launch these clinical studies, but we don't have a coordinated effort to address actually our knowledge bottlenecks right now. So that once we have these tests, we will then have the information how to deploy them most effectively. And then the national strategy could also be in terms of how do we promote the Canadian supply chains? And I think maybe Health Canada does a bit of that, but it's a relatively passive approach where we basically invite people, companies to come. But we're kind of in an all-out battle with this virus, and I think it is a more proactive way of also trying to build up the capacity here in Canada. And where I think there is opportunities is that the rapid tests, the technology underlying these rapid tests is from the 80s. There's no IP protection. So we have the opportunity, for example, and that's one thing I'm advocating for, an open source type of effort, where we and I think Canada would actually have a great opportunity to take a leadership role in this, in making this initiative by developing a test based on known information that on one hand could reduce the cost potentially, and also could then benefit from the knowledge input from all the other countries and scientists around the world. And this could then help accelerate the development of these tests, and also actually have this mutual benefit and actually building up a bit the solidarity, which I think has been lacking by and large in the course of this virus, of this pandemic.
David, I want to thank you for joining us.
Well, thank you for having me, and hopefully things will get better as we move forward.
Let's hope so. David Junker is a professor and chair of the Biomedical Engineering Department at McGill University. While rapid and accurate COVID testing is critical to clamping down on spread, a key component of that is the capacity for lab testing. Christine Nielsen's the Chief Executive Officer of the Canadian Society for Medical Laboratory Science, and she joins us now. And Christine, you deal with technologists and their assistants and the lab techs. Are they under pressure to increase turnaround times?
Definitely, especially in a place like Ontario, where we've set some pretty aggressive targets for how many tests we'd like to have completed every day, and they're in the tens of thousands.
And why is Ontario so far behind? Is it just numbers?
I don't know if it's far behind. So Ontario has a different testing protocol than some of the other provinces. We also have some hotspots, as you're seeing. So one of the things we've heard from the beginning is that one of the keys to getting a handle on what's happening with COVID-19 is to do more testing. It's also the volume, right? We have a huge number of people in Ontario.
Yeah, I can understand that, but you mentioned they have a different testing protocol. What makes it different than, say, Quebec or Alberta?
I think one of the things that even the premiers pointed out is that we have different medical officers of health, and we have a lot of them in Ontario. And then you have a place like British Columbia where there's a single medical officer of health making decisions for all of British Columbia. So that's some of the differences you're seeing. But we're also seeing differences in school boards, right? So Ontario has multiple school boards that are making different decisions about when children need to have tests to return to school if they've had a runny nose or a cold. So, and we also have some employers that are asking for COVID negative tests. A good example is if somebody's going in for elective surgery, in some cases, they've asked you to produce 2 COVID negative tests, and the same with access to long-term care facilities. So it's not just not just the number of people, it's that we've got all these external influences asking for you to prove you are COVID negative before you have access to something. And that's not happening in every province, to my knowledge.
Does Canada have the capacity for testing right now? Like, does the capacity match the demand?
The capacity for all of Canada does not match the demand. And that's because of a shortage of personnel, as well as in some cases equipment. So the test itself, the Pcr test for our favorite nasopharyngeal swab, takes about 4 to 5 hours per test. And that's why, from the beginning, from February or March, everyone's been looking for a point of care rapid test that can help speed up that turnaround. And there's lots under review right now by Health Canada, and some that have been approved for coming into Canada, which should help because that allows somebody who's not a medical technologist to use the device. We always ask that that's overseen by a lab to make sure that the device is doing what it needs to do. It's both specific and sensitive, so it's finding low viral loads. And that's the testing that's happening right now with all those devices, is they're busy making sure that the device is recovering low viral loads.
What hurdles are in Canada's way to increasing testing numbers?
So some people would say the regulatory framework in Canada is a hurdle, but as a citizen of Canada, I find it's one of the most reassuring things. I think about some of the things, flashback, 8 months ago, there's certain drugs you can buy in the United States over the counter. You just walk into a pharmacy and ask for it. And in Canada, we have regulations around some of those controlled substances that things have to be tested as helpful to the population, not harmful. So there's always been different regulatory standards around the globe, and Canada has some of the highest in the world. And I will say Health Canada's done a fabulous job of, I'm not going to say lightening the standards, but accelerating COVID-19 tests through. If you're on the website, there's pages and pages of documents that show that lots of tests are under review right now. They're coming from Korea, China, Germany. And so the scientists are making sure that those are doing what they need to do so that when we get a result, positive or negative, we can trust it. So I would say we're a little bit behind on demand. We also don't have enough people, the medical technologists and lab assistants, like you mentioned at the beginning, to do the jobs. And we, in some cases, don't have all the equipment, but we're getting there.
Christine Nielsen joining us in the Unpublished Cafe. She is the CEO of the Canadian Society for Medical Laboratory Science as we talk about rapid testing in Canada. And, we talked, you talked about the, I guess, the shortage where you could probably use more lab techs and assistants and people like that. Was that industry already in need of more people before the pandemic? And this is just exasperating?
Absolutely. So I would say my society's been in business since 1937, and one of the things we do is keep our eye on the health human resource needs, both for today and for tomorrow. So starting in 2000, we literally looked at the membership and said, my goodness, all the baby boomers are going to leave at the same time. Lots of people work in a pensioned environment, and it's a high-stress job. So we can predict the average member retires around 62. So we know what that number is. All you have to do is extrapolate data. And in 2000, we said the big problem is going to hit in 2015. And at that point, half of the eligibility was for our workers to retire. Now, we had the economic meltdown in 2008, so lots of people stayed on a little bit longer. But by 2020, which, we're here now, we've got at least 20% of the membership that can retire today.
Now we talk about the people. What about the labs, the tech, that kind of thing? Do we have enough of that, or is that also in demand?
the technology for sure is in demand. And one of the hard things, the way that hospitals are funded is they often don't get a lot of money for capital. And that's the equipment that we use to do our job. So we've all seen hospitals having capital campaigns. We need a new MRI. We need to buy new lab equipment. It's one of those things that's not usually thought of in advance by budgeting. And so that's something that likely needs to change. I will also say, though, during the pandemic, governments from across the country freed up those resources. And, we don't have to go through the same hurdles we usually do to get a new PCR equipment in the lab. They literally, just like Health Canada, expedited the system so that they could acquire what they needed. So despite it being a really regulated environment and, there's always rules in place, everyone has come together to say this needs to move faster, and how do we do that safely?
Would a rapid at-home test alleviate the pressure on your members?
So an at-home test causes me a little bit of stress, only because what if you come up positive? Who do you report that to, and how do we do contact tracing? So I'm not sure that at home is where we want those, but definitely in a place like, I know that there's great interest in putting them into some of the long-term care facilities and moving them into the rural areas of Canada. But the most important thing is that the device is accurate and that it's doing its job, and that we can contact trace so that we know that person A has device 1, and that when it comes up positive, here's how we'll track it back. It can help, but it does need to be accurate and specific.
Christine, I want to thank you for joining us.
Thank you.
Christine Nielsen is Chief Executive Officer of the Canadian Society for Medical Laboratory Science, which represents medical lab technicians and assistants. And that leads to our unpublished .vote question. Do you feel a rapid, highly accurate, and inexpensive COVID test will be available in Canada by the end of 2020? Yes, no, or unsure. You can log on and vote right now at unpublished.vote and have your voice heard. I want to thank Dr. Stephen Newmaster, genomics professor at the University of Guelph, David Junker, professor and chair of biomedical engineering at McGill University, and Christine Nielsen, CEO of the Canadian Society for Medical Laboratory Science. And I want to thank you for listening to the Unpublished Cafe. Stay safe. I'm Ed Hand.